Considered the external structures of the cell, let's move on to take a look at the cell envelope. This is the boundary layer of bacteria. The majority of bacteria have a cell envelope, and it lies outside of the cytoplasm and contains the contents of the cell. It's composed of 2 to 3 basic layers. All of them have the cell wall and the cell membrane around the cytoplasm, and then some bacteria have an outer cell membrane. Often the layers are tightly bonded together, and each layer performs a distinct function, but together, the end-all is that they act as a protective unit for the cell. We discussed in the last chapter a staining technique called Gram staining. This staining technique elucidates differences in the cell envelope structure. The differences that we see between gram-positive and gram-negative bacteria lie in the cell envelope, specifically in the peptidoglycan layer. Gram-positive bacteria have 2 layers, the cell wall, which is composed of peptidoglycan, and then the cytoplasmic membrane, whereas gram-negative bacteria have 3 layers. In addition to the cell wall and the cytoplasmic membrane, they also have an outer cell membrane. Here you can see the gram-positive bacteria with this thick layer of peptidoglycan, the brown layer, and then a thinner cell membrane. And on this side we see the gram-negative bacteria, where the brown peptidoglycan layer is much thinner and it has an outer cell membrane. This structure will help explain why we see gram-positive and gram-negative staining. Because all bacteria have the cell wall layer, let's take a look at that first. The cell wall helps determine the shape of the bacteria, and it provides really strong structural support. Most of these cell walls are rigid because of this peptidoglycan content. Glycans simply means sugar chains, and peptide, means to do with polypeptide chains. So peptidoglycan means we've got sugar chains and protein components. More specifically, we see these alternating glycan chains composed of G's and M's. The G is N-acetylglucosamine and M is N-acetylmuramic acid. So they alternate in these chains, and these chains are then held to each other by these peptide crosslinks. We can take a closer look at the arrangement here. The M units, or muramic acid units, are held together by an interbridge. This layer of peptidoglycan keeps the cells from rupturing because of changes in pressure due to osmosis. Bacteria live in a relatively dilute environment of solutes, and thus water is always diffusing into the cell. Many antibiotics target this cell wall structure. For example, penicillin and cephalosporins are effective because they target that peptide crossbridge in the peptidoglycan structure. It disrupts the integrity of the peptidoglycan layer, and thus cells are more likely to lyse or break apart. So essentially, penicillin and cephalosporins are disrupting cell wall structure so that the contents can no longer be maintained inside the cell. Now, enzymes in our tears and saliva called lysozyme act to break down the glycan chains, so the sugar subunits fall apart. This also causes the wall to break down. So this is one of our outer mantle components that protects us from invading bacteria. The gram-positive cell wall is much thicker than the gram-negative cell wall, the peptidoglycan layer. It's about 20 to 80 nanometers thick, and it's more of a homogeneous sheath of peptidoglycan that contains tightly bound acidic polysaccharides. The gram-negative cell wall, as you can see over here on the right, has a very thin layer of peptidoglycan. It's only about 1 to 3 nanometers in thickness. And outside of that, there's a periplasmic space surrounding the peptidoglycan. This thinner layer gives gram-negative bacteria a much greater flexibility. However, it makes them much more susceptible to lysis, or breaking apart. This is a great time for us to check out an animation on how gram staining works.
The gram stain is the most widely employed staining method in microbiology. It is a differential stain because it divides bacteria into 2 classes, gram-positive and gram-negative. In the first step of the gram stain procedure, cells from a fresh culture are transferred to a clean slide and allowed to dry. If the cells are on an agar plate, they should first be transferred to a liquid medium for dilution. The cells should form a thin, barely visible film. This can be achieved by smearing cells obtained from the surface of an agar medium or from a liquid culture. Fresh cultures must be used because as cells age, they lose their ability to retain the stain. The cells are then fixed to the slide by passing slightly above the flame of a Bunsen burner. After passing above the flame, the slide should feel warm when touched to the back of the hand, but should not be too hot. The fixed cells are then stained with the basic dye, crystal violet, for 30 to 40 seconds. The slide is then rinsed with water to remove excess stain. At this point, all cells appear purple under the microscope. Next, a solution of Graham's iodine is added and retained on the slide for about 1 minute. The iodine combines with the crystal violet to form a di-iodine complex, thereby decreasing its solubility within the cell. At this point, the cells still appear purple. The cells are then decolorized by washing with ethanol or acetone. This is the differential step. Gram-positive bacteria retain the crystal violet, whereas gram-negative bacteria do not. The ethanol or acetone should be added drop-wise, with the slide tilted at an angle until the drop coming off the edge of the slide just starts to become colorless. Even gram-positive cells can lose the crystal violet iodine complex during prolonged excessive decoloration. Excess ethanol is then washed off with water. When viewed under the microscope, gram-positive cells appear purple and gram-negative cells are colorless. Finally, the rinsed cells are covered with the counterstain safranin for 20 to 30 seconds. This stains the gram-negative bacteria pink. After rinsing with water, the slide is dried with filter paper. When viewed microscopically, the gram-positive bacteria are purple and the gram-negative bacteria are pink. Generally, the gram stain correlates with the cell wall structure among the bacteria. The ethanol is thought to shrink the thick peptidoglycan in gram-positive cells, thus retaining the dye. The thick, dehydrated peptidoglycan layer of gram-positive bacteria appears to be a permeability barrier, preventing the loss of the crystal violet iodine complex. In contrast, the peptidoglycan in gram-negative bacteria is very thin and has large pores. Ethanol may extract lipids and increase the porosity, thus removing the crystal violet iodine complex.
And here's a great figure to summarize the stages of gram staining. Here we can see what happens in the crystal violet stage. Each of the cells maintains this purple coloration because both cell walls will affix the dye. In Graham's iodine staining, we'll see that the gram-positive bacteria will have the dye complex trapped in its cell wall, the thick peptidoglycan there, and then no effect of iodine on the gram-negative. However, both still appear purple. Once we do the alcohol rinse, we see that the crystals remain in the cell wall, and thus the gram-positive bacteria will still be purple, and the dye is washed out of the gram-negative cell as the peptidoglycan layer is thin and protected by the outer membrane. Finally, we have the safranin stage, and in this stage we're simply counterstaining the gram-negative bacteria. So gram-positive appear purple and gram-negative appear pink. In addition to gram-positive and gram-negative bacteria, there are some bacteria that have non-typical cell walls. Some of them aren't characterized as either gram-positive or gram-negative. Indeed, some don't have a cell wall at all. Archaea, for example, are unusual and chemically distinct from the general bacteria. The archaeans are a more ancient and primitive line of prokaryotes. Some of their cell walls are composed entirely of polysaccharides, and others are composed entirely of proteins. Now, mycoplasmas and a very few archaea lack a cell wall entirely. Their membrane is stabilized by sterols and is resistant to lysis. They're very small bacteria, 0.1 to 0.05 microns, and they range in shape from filamentous to coccus. They have important medical species, the Mycoplasma pneumonia, for example. is an atypical form of pneumonia where the bacterial cells actually adhere to the epithelial cells of the lungs. Another non-typical cell wall is seen in Mycobacterium and then Nocardia. They're unique types of lipids in these cell walls. Both of these contain peptidoglycan and they stain gram-positive, but the cell wall is actually composed of long-chain fatty acids, unique types of lipids. They're waxy, and so they're resistant to chemicals and dyes. They contain mycolic acid, which is the waxy substance. In order to identify these bacteria, we use an acid-fast stain in which the hot carbofuchsin dye holds fast. That is, it's not washed off by the acid alcohol, hence the name acid and fast. Pathogens that have this sort of structure in the cell walls are tuberculosis, leprosy, and some other opportunistic wound infections. Let's have a quick concept check. You've isolated a bacterium from your skin. Chemical analysis shows that it contains proteins, peptidoglycan, lipids, DNA, and acidic polysaccharides. What sort of bacteria is this? We're really looking to see if it's gram-positive or gram-negative. What's the one thing that gives it away here? They all have peptidoglycan, they all have lipids and DNA. The key here are the acidic polysaccharides. These are only found on the membrane of gram-positive bacteria. So now that we've considered differences in the cell wall structure between gram-positive and gram-negative bacteria, let's take a look at the outer membrane. The outer membrane is similar to the cell membrane, except it contains specialized polysaccharides and proteins. Now you'll recall that gram-positive bacteria don't have this outer membrane, so we're only talking gram-negative here. The uppermost layer contains lipopolysaccharides, or LPS, the lipid portion of which is referred to as an endotoxin because it stimulates fever and shock reactions in gram-negative infections like typhoid fever and meningitis. So the patient in this case, it goes into shock and fever because of interactions that the lipid portion on this outer cell membrane has with the human body. The polysaccharide portion is composed of chains that extend off of the cell surface. You can see those in green here. The innermost layer, or the phospholipid layer, is anchored by lipoproteins to the peptidoglycan layer below, so it's in tight association. This outer membrane serves as a partial chemical sieve, where only relatively small molecules can penetrate. These small molecules are allowed to penetrate through the porins. The porins have variable size, which can be controlled by the cell to disallow or allow molecules of a variety of different sizes. This porin size is the gram-negative bacteria's main defense against some of the antibiotics we use for treatment. So this is one of the reasons that gram-negative infections are much more challenging to treat. So now let's take a look at the cell membrane structure, so the inner membrane. This membrane is also known as the cytoplasmic membrane. It's very thin and it contains primarily phospholipids and proteins, just like a eukaryotic cell membrane. The exceptions here are in mycoplasmas and archaea. They have a slightly different structure, which we'll examine later. The cell membrane also provides a site for functions such as energy reactions and nutrient processing and synthesis. Most enzymes of respiration and photosynthesis reside in the cell membrane because they have no mitochondria, so any of these reactions have to happen in the cell membrane. We'll also find enzymes that help synthesize the macromolecules that are embedded in the membrane. Now, water and uncharged molecules can diffuse across this cell membrane unaided. Because this membrane is selectively permeable, it has embedded proteins that help regulate the transport of materials in and out of the cell. In addition to bringing molecules in, the membrane allows export of molecules or discharge of metabolic products through the process of secretion. So now that we've considered all three layers of the cell envelope, let's look at some differences in their structure. The outer membrane is an extra barrier in gram-negative bacteria. This makes them impervious to some of those antimicrobial chemicals or some antibiotics. Because of this impervious layer, it's often more difficult to inhibit or kill gram-negative bacteria than it is to kill gram-positive bacteria. In addition, the envelope can interact with human tissues and cause disease in itself. This is the case with Corynebacterium diphtheriae, the one that causes diphtheria, and Streptococcus pyogenes, the one that causes strep throat. Proteins attached to the outer portion of the cell wall in these bacteria have toxic properties in their interactions with the human immune system. So this brings us to the end of our section on the cell envelope. Next, we'll embark on an exploration of the internal structures in bacteria.